We're going to do it now. Another organelle we should look at will be the, you know what, I'm going to do the nucleus next. Here, the nucleus. Notice that the nucleus and the rough ER are kind of hooked together. The nuclear envelope, which is in purple up here, the nuclear envelope is generated by the rough ER. Right after a cell divides, the rough ER kind of surrounds the DNA and it produces the nuclear envelope. All right, now we've got a picture of that in the book.
The nuclear envelope is produced by the rough ER?
Correct. All right, here we go. Let me see if I can get all of this. This is on page 99. And then you got it look better than that.
You said it was combined to the what again?
Say that again, please.
It was combined to
The nuclear envelope is made out of the rough ER.
right, but you said it was combined to something else.
I don't know, what did I say?
The ER, the rough ER.
It's it's connected to the rough ER.
Okay.
I don't know if that's what if I said anything else or not. All right, that picture doesn't show up really great. But here, in this diagram, you can see that the nuclear envelope has two membranes. That's because it came from the ER, which has two membranes. It's kind of snugged up around it. But there's lots of holes in the nuclear envelope. Those are called the nuclear pores. And so they allow things to go in or out. This is the way stuff gets in or out of the nucleus. All right, well, let's see then. Here in this lighter area here, it's over here also, this lighter area. It looks a lot better in the book. But the lighter area is called chromatin. That's DNA. And of course, that's where we find DNA. It's going to be inside the nucleus. So here it is. This pale, lighter region is the chromatin. The dark region here, this big dark blob, here they show in both diagram and micrograph, is the nucleolus. That's labeled for you also. And the nucleolus is made primarily of RNA. This is where the ribosomes are made. Ribosomes are mostly RNA. They're both RNA and protein. But they're constructed or generated here in the nucleolus. So the ribosomes have to get out, because where do we find them working? They're sitting on the rough ER. So they got to get away from the nucleolus, they got to get out into the cytoplasm, and then go stick down on a piece of ER somewhere. All right? So just realize that ribosomes are generated in the nucleolus. All right, now let's see. All right, any questions? How are they going to get out of the nucleus? Through what? The pores, the nuclear pores. Okay, nuclear pores also let messenger RNA, which is a little shuttle that's going to carry instructions out. We'll see that in another place. So there's other things that have to go in and out through those nuclear pores. All right, so much for that. Now, anybody have a question on nuclear envelope? Well, what else can we look at? All right, let's see, some more membrane stuff in the cell. The next item we're going to look at is the Golgi apparatus, which is here in this diagram. Right here.
What is it?
Golgi apparatus. Let's see. There you go.
Okay.
Golgi.
Right here. But I can fix it. Okay, Golgi apparatus. It was named after an Italian guy named Golgi. He saw it the first time, I guess. All right. So the Golgi apparatus is an interesting organelle. It forms from the endoplasmic reticulum on one side, forms from the rough ER, and it comes apart on the other side. So it's forming on one side and coming to pieces on the other. The function of the Golgi apparatus is to process, package, and release secretions. Process, package, and release secretions. So, see, remember, what is it that's making the enzymes, the proteins for those little organelles?
Ribosomes.
They're making little baby enzymes. They're not grown up, that is to say. They're not mature enzymes ready to work, but they're just little baby enzymes. And so the ribosomes make those, stick them into the ER. The ER ships them off to some place where a Golgi apparatus is forming. And the little baby enzymes are pinched off into vesicles that become a part of the Golgi apparatus. Then inside the Golgi apparatus, I don't know what turns everything on, but there's a little machinery in there that starts working on those baby proteins and turns them into mature proteins. And then when they're mature proteins, they pinch off into vesicles. Here's the term secretory vesicle. And the vesicles may come here and fuse onto the cell membrane, and all the little, whatever was in the vesicle gets released outside the cell. Now, a good example would be digestive enzymes. Your pancreas, the cells of your pancreas, make digestive enzymes. And the cells, once they have finished making those enzymes, the Golgi apparatus sends little vesicles out to the cell membrane, and poof, they go out into the pancreatic duct and then go to your intestines, where they can digest your food. So that's what goes on. See, our cells are really good things. Matter of fact, if you've had anything to eat yet, that's what they're doing right now. All right. Now, let me see. There's another picture, I think, in your book, maybe. Let me see. I'm looking for a good Golgi picture. It's right over here. All right, here's that. Here you go. Let's look at this one. Okay, well, this doesn't show it really that great, but here's a rough ER, and it doesn't show vesicles, but they're vesicles that pinch off the rough ER, and they come and fuse onto, are fused together and make one of these little pockets that make up the Golgi. And they break it up. This shows it really good. They break it up on this side, and these are the little secretions in here, and here's being released. There it goes. But as this side disappears because it's all being pinched off into vesicles, new ones are coming on here, and so the thing is constantly progressing, constantly in action. All right. Those secretions that are shown here are like the digestive enzymes that get sent out of the cell, and they'll go to work on your food. There are other vesicles that come off of the Golgi that stay inside the cell. And some of these are called lysosomes. Here's the picture your book has of lysosomes. Makes it look like they're great big things. Lysosomes are really very tiny. And this is magnified 14,000 times or so. But lysosomes come off of the Golgi. They contain digestive enzymes, but they stay in the cytoplasm of the cell. The best example that we have of that, I think, are the white blood cells. They contain lysosomes. And when a white blood cell, white blood cells patrol through your system all the time, and if there's any bacteria, the white blood cell will phagocytize it. That is to say, it will eat it. So the bacterium gets into the white blood cell, and the lysosome comes up and fuses with that and digests the bacteria. So it's digesting things that don't belong anymore, or never belonged at all. lysosomes in all of our cells recycle old cell parts. That is, for instance, if there are ribosomes that have been working really hard and now they're just worn out and old, they're not working very well anymore for whatever reason, a piece of them fell off or something happened, then they will be, lysosomes could digest those. Or old mitochondria, for instance, that other old organelles in the cell that aren't working properly could be digested by these lysosomes. All right, so anyway, that's what they're for. Recycling inside the cell. Your book also mentions peroxisomes down here. No pictures, but it's got the word. They're also tiny little organelles that, in which certain chemical reactions occur in the cell. So it's a little membrane-bound vesicle where chemical reactions occur inside. The reactions that occur in peroxisomes make a very bad substance. They're the byproduct. They make hydrogen peroxide, which is a destructive molecule inside cells. So peroxisomes also contain the enzyme that destroys the hydrogen peroxide. Now, you see that in your book? Maybe you can see it over here. There, no, where is it? Catalase, there it is. Peroxisomes contain this enzyme catalase that will tear up about 250,000 hydrogen peroxide molecules per second. Does that impress you? That impresses me. Can you count to 250,000? I mean, it would take a while to count to 250,000, but it does that operation that many hydrogen peroxides every second. So that means there's a lot of chemical activity going on. All right. So much for the peroxisomes, and that takes care of most of the, well, really all of these membrane-bound organelles that have to do with the endoplasmic reticulum or that come off of the endoplasmic reticulum. But now we have one other membrane-surrounded organelle that I need to mention, and that is the mitochondrion. Okay, so here we go with mitochondria. Mitochondria, let me see, I have two membranes. Not just one, but two. The book doesn't have a great picture of it, but this is okay. Here, the diagram, you can kind of tell things, or at least see what they're trying to show in the diagram better really than you can here. But you've got an outer entire membrane that just encloses the whole thing, and then the inner membrane folds back and forth, making these these folds in here called cristae. So you can't really tell what here's pointing at this crista right here. All of these plate-like things going across here are the cristae. And it's on the inside of the mitochondrion, and particularly at the cristae, that's where a lot of adenosine triphosphate is made. Ooh, that's ATP. I got the word written up here already, but it didn't show up very well. Oh, it doesn't show up at all. Forgot about that. ATP, adenosine triphosphate. So, let's see. I don't know, that word will be in your book.
On page 94.
Oh, is it? Okay. Oh, yes, in bold print. Adenosine triphosphate. Okay, ATP. All right, so, and all of our cells use ATP. They're using ATP right now. Every one of your cells is using that stuff. And the mitochondria are churning it out. And how do I know that's so? Because you're breathing. When you breathe in, you're taking that oxygen in to the mitochondria so they can do that process. Let's see, I just heard that we use 200